Good day, students. In this lecture, we are going to talk about mitosis again. And in the previous lecture, we were talking about mitosis. And in mitosis, we were talking about karyokinesis. And I have told you that karyokinesis is further divided into four steps: prophase, metaphase, anaphase, and telophase. Now we have discussed till prophase. So now we are going to start from the second step of karyokinesis of mitosis. So I should write. We are talking about mitosis. So the second step of the karyokinesis after prophase is the metaphase. We have talked earlier about the prophase. It is the longest phase of cell division, mitosis. Now we are going to talk about metaphase. Now, if we talk about metaphase, what are the main changes that occur during this phase? The one major activity or the one major process that takes place during this phase is that the chromosomes which are attached to the spindle fiber they get arranged at the center or equator of the cell, and this forms the metaphase plate. So the major characteristic change that occurs during this phase is that the chromosomes get arranged at the equator. And this forms and this is called the metaphase plate. So in one word, we can say that during metaphase stage, formation of metaphase plate occurs. Remember, equator means center of the cell. One more point that we must remember about metaphase is that during this phase, the chromosomes are shortest and the thickest. So they have condensed to their maximum, so they appear very shortest and thickest, and it is the most suitable phase to study the chromosomes in terms of number, in terms of their size, shape. So overall study of the chromosomes is best done at the metaphase stage of the cell cycle, metaphase stage of mitosis. So chromosomes are shortest and thickest during the metaphase stage. Best stage to study the chromosomes. In terms of number, shape, as well as, so we can say the overall study of chromosomes can be done at this. If we represent metaphase stage diagrammatically, say for example, if this is the cell, and we are representing the spindle fiber. Remember, there are two types of fibers, the continuous fibers and discontinuous fibers. The continuous fibers extend from one pole to the other pole, whereas the discontinuous fibers extend from one pole to other pole, but they are attached to the chromosome's centromere. So we can see in this diagram that the chromosomes are arranged in the center of the cell to form the equator or metaphase plate. This is called as the metaphase. Remember one thing, that the chromosomes are attached to the spindle fibers at their centromeres, and I have said that there are two types of fibers, one depicted by the continuous orange yellow line, dark yellow line, and the discontinuous sorry the discontinuous, which is represented by the continuous dark yellow line, whereas the continuous, which is represented by the dotted dark yellow line. The discontinuous fibers are extending from one pole to the other pole, but they are attached to the chromosomes. But the continuous spindle fibers, representing by the dotted lines, extend from one pole to the other without being attached to the centromeres, chromosomes centromere. Now, after metaphase, the third step of the karyokinesis is anaphase. The major changes that occur during anaphase is that in this phase, centromere, which is joining the two sister chromatids, divides into two to form two daughter chromosomes. So, the two daughter sister chromatids, which are joined at the centromere, are now separated from each other, and now they behave as the individual chromosomes. But the respective centromeres of the separated chromosomes remains attached to the spindle fibers, and now these spindle fibers start contracting, so these newly formed chromosomes start moving to their respective poles and they become shortened, so they move towards the respective poles. Now, as I told you that the centromere position can be different, and this determines the shape of the chromosome. So, during the anaphase stage, we can clearly see the shape of the chromosome easily depending upon the location of the centromere. Remember one thing, if you want to study if you want to do whole study of the chromosome, then we should do it during the metaphase stage. But if you want to study the shape of the chromosome only, then you should select anaphase stage of the cell division. So we should write the major changes. And besides this, the spindle fibers shorten or contract. Anaphase is the shortest phase of mitosis. Remember, prophase is the longest phase, whereas anaphase is the shortest phase. These are some of the characteristic features that should be remembered. Chromosomes appear in different shapes, and therefore it is the suitable stage for studying chromosome shape, and the different shapes are depending upon the location of the centromere, and the different shapes are V-shaped due to metacentric centromere. Metacentric means that the centromere is located in the center, exact center. Therefore, the chromosome appears in as V. The second shape is L-shaped. Here, the centromere is submetacentric in position. Submetacentric centromere. Now, here the centromere is located somewhat at not at this exact center but away from center, little away from center. Therefore, it leads to formation of two unequal arms. One is slightly longer, another is slightly shorter, and they form a L shape. The third type is J shape. Here, the centromere is acrocentric, which means that the centromere is located far away from the center but near the terminal end. So, one arm, it leads to formation of two unequal arms. One is quite large and the other arm is very short, and it takes a J shape. And the last shape is I shape. Here the centromere is terminal in position, which is called as telocentric centromere or terminal. Since the centromere is present at one of the end, it forms a single arm and therefore it takes a shape of an I, small I. The alphabet I, not the I or the. So during anaphase, as the anaphase proceeds towards end, a constriction starts developing. It means that cytokinesis is also starting by the end of anaphase. These are the daughter chromosomes. You can clearly see the difference between the daughter chromosomes and the parent chromosome, that the parent chromosome was containing two chromatids, whereas the daughter chromosomes are containing single chromatids. So, basically, the daughter chromatids are formed by the separation of the two chromatids of the chromosome. So, these ensures that the two daughter cells receive same number of chromosomes. This is representing anaphase. As we can see that a constriction has started developing at the end of anaphase, which marks the start of cytokinesis also. Remember, we are talking right now about the animal cell divided by mitosis. We will see the difference by which the plant cell divides by mitosis. In the end, we will discuss that differences also. So, this is all for today's lecture. In the next topic, we are going to study the remaining stages, that is telophase and the cytokinesis. Have a good day.